major research interest of our lab investigated in this paper is how learning and memory deficits are initiated in Alzheimer's disease. Since Alzheimer's description of the AD patient August Dieter, scientists have been trying to determine what triggers the inability to form new memories early in the progression of Alzheimer's disease. The current dogma is that in places like the hippocampus, a major site of learning and memory in the brain, synaptic connections among excitatory neurons, visualized as postsynaptic dendritic spines, are excessively eliminated in Alzheimer's disease, causing memory deficits. Yet little work has been done to substantiate this claim. Given studies of motor learning show that new synapse assembly is highly correlated with learning and required for it, we hypothesized that learning deficits in AD might arise from an inability to form new spines, not the destruction of pre-existing ones as is currently believed. To mimic early stages of Alzheimer's disease, we used amyloid beta peptides. Amyloid beta peptide oligomers are enriched in plaques in AD patients. They're the earliest known markers of Alzheimer's disease. They're genetically implicated in AD and highly synaptotoxic. We find following exposure of A-beta oligomers to dendritic spines in live imaging studies that new spine assembly is specifically inhibited, consistent with our hypothesis. We gain mechanistic insight in how A-beta inhibits synapse assembly by studies of the NgR receptor family, a family of cell surface receptors we previously showed restrict synapse assembly developmentally. Our experiments revealed that NgRs mediate A-beta's inhibition of synapse assembly, A-beta's inhibition of synaptic plasticity, specifically LTP, and that NgRs bind A-beta oligomers on neurons. To localize A-beta NgR signaling on neurons, we imaged live two spectrally distinct fluorescent sensors that were expressed in neurons. One measuring RhoA activity, since RhoA is a known NgR signaling mediator, and one measuring calcium activity, since calcium dysfunction is a hallmark feature of A-beta pathology. Imaging studies of neurons expressing these sensors revealed, following A-beta exposures, that NgR RhoA signaling is specifically activated along the dendritic shaft, and this NgR signaling activation inhibits calcium activity in this domain.
To determine what is the calcium target of A-beta NgR signaling, we screened pharmacological blockers of calcium regulators, identified inhibitors of T-type calcium channels as blockers of calcium signaling in dendrites. We hypothesized that A-beta NgR signaling might target T-type channels. To address this hypothesis, we recorded T-type currents from neurons, revealing A-beta inhibits T-type currents. This inhibition was NgR and Rho kinase dependent.
To address the in vivo relevance of the proposed A-beta NgR signaling pathway, we injected A-beta oligomers into the ventricles of control mice and found that A-beta reduces novel object recognition learning, spine density, and recordings of T-type currents. In contrast, these deficits were rescued in NgR family knockout mice that were also injected with A-beta.
This study highlights the possibility that early learning deficits in Alzheimer's disease may be mediated by the inability to assemble new spines during learning, a mechanism that involves NgRs and T-type channels. Thank you.